When you think about how we develop, how a single fertilized egg turns into a fully formed person or an animal, we usually picture this incredibly peaceful, harmonious process, like a microscopic orchestra tuning up together to play a symphony.
Right, yeah, it sounds very elegant.
Exactly, elegant. But I want you to imagine something entirely different today. I want you to imagine a microscopic Hunger Games happening inside every mammalian embryo right at the dawn of its development. Like a brutal, enclosed arena where only the absolute fittest cells survive to build the body, and the rest. They are systematically, ruthlessly eliminated. Welcome to today's deep dive.
It is a really phenomenal image, and frankly, the reality of embryonic development is even more fascinating than the metaphor. We are looking at a deeply embedded biological process where cells are literally judging the fitness of their neighbors.
Wow.
Yeah, they decide who gets to contribute to the final organism and, who has to be discarded.
And our mission today is to figure out the actual mechanics of this microscopic battle royale. We're diving into a really compelling 2026 paper from Nature Cell Biology that explores a phenomenon called developmental cell competition.
It's a fantastic paper.
It really is. We're going to unpack the underlying machinery of how these tiny cells judge each other, the strict timeline of when this deadly competition happens, and why this violent purge is actually crucial for our very existence.
To even begin to understand this, though, we have to look at the battlefield itself. Studying early mammalian embryos, say a mouse embryo at day 5 or 6 of development, is historically a massive technical challenge.
This is so tiny, right?
Exactly. They're incredibly small, they develop inside the mother, and observing their real-time cellular interactions without disrupting them is, well, it's notoriously difficult. So the researchers in this paper used a 3D stem cell model called a gastruloid.
Okay, let's unpack this. What exactly is a gastruloid? Sounds a bit like, I don't know, science fiction.
I mean, it kind of is. It's essentially an aggregate of embryonic stem cells grown in a laboratory. If you take a specific number of these stem cells and place them together in a 3D suspension, over the course of about 5 days, they self-organize.
Wait, they just do it on their own?
Yeah, they just do it. They elongate, they establish an axis, and they actually mimic the highly complex early stages of embryonic development. It provides researchers with this transparent, observable window into a phase of life that is otherwise completely hidden from us.
I want you listening to really visualize this. Picture a tiny self-assembling biological sphere floating in a lab, acting out the first days of life. So we have our arena, this 3D gastruloid model. But to study a competition, you need an uneven playing field, right? How do the researchers actually trigger a fight in the lab?
They use genetics to engineer an underdog and a champion. The researchers started with a population of normal, healthy stem cells. We call these wild-type cells. And to keep track of them under a microscope, they genetically labeled them with a harmless fluorescent marker that glows green.
Okay, so normal cells are green.
Right. Then they introduced a mutant subclone of cells, and they labeled those to glow magenta.
So we've got green versus magenta, but what makes the magenta cells mutants?
They had one specific crucial gene completely deleted from their DNA. The gene is called TRP53, which produces a protein known as P53.
Hold on, I've heard of P53. Isn't that usually talked about in the context of cancer? Like, if your body loses P53, you are in serious trouble.
That is the exact context most people know it from. P53 is famously called the guardian of the genome. In an adult human, its job is to monitor cells for DNA damage. If a cell is broken, P53 stops it from replicating.
So it's like the quality assurance inspector.
Exactly, and when you lose P53 function in an adult, tumors can grow unchecked. But in this specific early embryonic environment, the rules are totally different. Lacking P53 doesn't just make these magenta cells grow wildly, it fundamentally alters their social behavior. It turns them into what the researchers term supercompetitors.
Supercompetitors. And the sheer dominance of these supercompetitors is what really stopped me in my tracks while reading the source material. I want you to picture the math on this. The scientists built a gastruloid, starting with 150 normal green cells. Then they dropped in just 2 of the magenta P53 mutant cells.
Yeah, just 2.
Just 2. And those 2 supercompetitors completely hijacked the entire structure. The normal cells essentially became losers, their growth stalled out completely, and the mutants took over the entire population.
It's a staggeringly asymmetric victory, isn't it? A little over 1% of the starting population is enough to completely suppress and replace the other 99%.
But I have to push back here, because when I hear that one cell type outgrows another, my immediate thought is just, well, it's a real estate problem. If you put 2 types of fast-growing cells in a confined space, Isn't it just a matter of the mutants multiplying faster and starving the normal cells out of room and food?
It's a highly logical assumption, and it actually highlights why using a 3D gastruloid is so important compared to older methods. If you grow these exact same two cell types flatly on a standard 2D Petri dish, competition only occurs when the dish reaches 100% confluence.
Confluence meaning they cover the whole bottom of the dish.
Right, meaning they physically run out of room and bump into the plastic walls. They only fight when they literally have nowhere else to go.
Like a crowded dance floor where people are just accidentally elbowed off the edge.
That's a perfect analogy. But in the 3D gastruloid, the architecture allows for expansion in all directions. The cells aren't just running out of physical space. The supercompetitor is actively, deliberately inducing the normal cells to stop growing or die. It is an active, non-autonomous quality control system.
Non-autonomous meaning the normal cells wouldn't die on their own.
Yeah.
They are receiving a targeted execution order from their neighbors. And here's where it gets really interesting. The overall size of the gastruloid doesn't actually shrink when all these normal cells are killed off. It's not like the structure collapses.
No, not at all, because the p53 mutant supercompetitors engage in something called compensatory growth. As they trigger the normal cells to die, the mutants rapidly expand their own volume to perfectly fill the microscopic gaps left behind in the tissue.
Oh, wow.
Yeah, they maintain the overall physical architecture and size of the gastruloid the whole time.
It's terrifyingly efficient. It's like an invading army that doesn't just conquer a city, but immediately moves into the houses, waters the lawns, and keeps the plumbing running so that from the outside, the city looks completely unchanged.
Yeah. The structural integrity is maintained even as the original inhabitants are completely replaced.
Okay, let's do a quick recap for you listening just to lock this first part in. Bullet 1, we have a 3D model called a gastruloid. Bullet 2, mutant cells lacking the p53 gene become supercompetitors. Bullet 3, just 2 of these can wipe out 150 normal cells. And bullet 4, they perfectly replace the dead cells without ruining the structure.
That's a great summary.
So, if they aren't just shoving each other off a cliff for space, how exactly are the mutants delivering this execution order? What are the actual weapons being deployed on this cellular battlefield?
Well, the researchers had to play detective and systematically test the environment to rule out the usual suspects. The first hypothesis is it a chemical weapon?
Right, like poison gas.
Exactly. Are the supercompetitors secreting a toxic protein into the fluid around them, acting like a long-range missile? To test this, they used microcavity plates. Imagine a tiny apartment building where the normal cells live in one open-top room, and the supercompetitors live in the adjacent rooms. The fluid overhead is shared, but the cells themselves cannot physically touch.
And if it was a secreted toxin, the normal cells would still die because the poison would just drift over in the fluid.
But they didn't. The normal cells grew perfectly fine. This proved the death signal is not a long-range chemical broadcast. It absolutely requires short-range, direct physical membrane-to-membrane contact.
Hand-to-hand combat. Okay, so what about mechanical crushing? Could the mutants just be flexing and physically squishing the normal cells until they pop?
They tested that by manipulating the mechanical environment. The scientists built gastruloids of drastically different sizes, some tiny ones starting with 75 cells and massive ones starting with 600 cells.
And what did that show?
Well, if mechanical crushing was the weapon, the intensity of the competition should change based on the density and pressure of the structure. But the elimination rate was identical across all sizes. They went a step further and measured a protein called YAP.
What does YAP do?
YAP is a mechanosensor inside the cell. It's like the cell's internal pressure gauge. When a cell gets physically squeezed or compressed, YAP proteins are literally shoved into the cell's nucleus to turn on stress response genes.
Oh, clever.
Yeah, so the researchers checked the nuclei of the normal cells, and there was no YAP accumulation. The normal cells were completely unaware of any mechanical pressure.
So no poison gas and no physical crushing. What about starvation? Did the mutants just steal all the nutrients?
They dropped the glucose availability in the surrounding fluid by 8-fold. 8 times less food. If it was a fight for food, severely restricting the calories should have changed the dynamic, perhaps slowing the mutants down. It didn't alter the competition at all.
So the normal cells aren't poisoned, they aren't squished, and they aren't starved. What is actually killing them?
Yeah.
I want us to go step by step here so we catch every detail.
Okay, step by step. The normal cells are driven to commit a highly orchestrated form of cellular suicide known as mitochondrial apoptosis.
Let's break that down. How does a touch from a mutant cell force a normal cell's mitochondria to initiate suicide?
When the membrane of the supercompetitor makes direct physical contact with the normal cell, it triggers a localized stress cascade. This signal travels inward to the normal cell's mitochondria. Now, mitochondria are the powerhouses of the cell, but they also act as a biological vault.
A vault. I like that visual.
Right. And inside that vault is a molecule called cytochrome C. The stress signal from the mutant causes the mitochondrial walls to become porous. The vault opens, essentially. The cytochrome C leaks out into the main body of the cell, and its release acts as an irreversible self-destruct sequence.
Like flipping the big red switch.
Exactly. It activates enzymes that quietly and methodically dismantle the normal cell from the inside out.
I do want to bring up one specific detail from the source material because it sounds incredibly dramatic. There's a mention of phagocytosis cells eating each other. Are the mutants literally eating the normal cells alive?
I know, the imagery is compelling, but it's really important to clarify the sequence of events here. The researchers did note that about 70% of the cells in the environment had engulfed fragments of their dead neighbors. But this is not the cause of death. It is just the cleanup crew arriving after the fact.
So it's not a microscopic Pac-Man situation where they're chomping on healthy cells?
No, not at all. When a cell undergoes apoptosis, it neatly packages its remains into little biological debris bags. The surrounding cells then engulf these bags to recycle the nutrients and prevent inflammation in the tissue. The crucial finding here was that both the normal cells and the mutant cells participated equally in this cleanup.
Okay, so the mutants aren't eating healthy cells alive. They are just helping sweep up the remains of the cells they previously forced to self-destruct.
Exactly, just standard cellular debris clearance.
All right, time for another quick summary check. We've ruled out long-range chemicals, physical squishing, and starvation. The weapon is direct contact that causes the normal cell's mitochondria to leak cytochrome C, triggering internal suicide. And the eating part is just garbage collection.
You nailed it.
But the source material highlights a massive caveat to all of this. The supercompetitors are not omnipotent. They can't just strike whenever they want. The arena has strict operating hours.
The temporal window. This is perhaps the most elegant discovery in the entire paper. By monitoring these gastruloids hour by hour, the researchers realized that this brutal competition only occurs during a highly specific time frame, between 48 and 96 hours of the gastruloid's development.
Which mirrors embryonic days 5.5 to 7.5, right? So what is biologically special about that specific 48-hour stretch?
It corresponds to a massive transition in the embryo's life. The cells are moving out of a state called primed pluripotency. Think of primed pluripotency as a waiting room. The cells have the potential to become bone, brain, or muscle, but they haven't received their assignments yet.
Okay, I'm picturing the waiting room.
The 48 to 96-hour window is when they leave the waiting room and enter early gastrulation. That's the phase where they actively commit to forming the foundational structural layers of the body.
But I'm curious how they prove the competition is truly locked to that exact developmental stage. Couldn't it just be that it takes the mutants, 48 hours to multiply enough to become a threat?
That's exactly the alternative theory they needed to test. And to do it, they performed what are called heterochronic shifts. Because these gastruloids are built in a lab, they are modular. You can pull them apart and mix cells from completely different developmental timelines. They basically created time-traveling cells.
Wait, really? So taking a cell from the future and dropping it into the past?
Yes. If you take a mutant supercompetitor from the 96-hour stage, so it has aged past the competition window, and you drop it into a 48-hour gastruloid full of young normal cells, the supercompetitor is utterly powerless. It cannot trigger the apoptosis cascade.
Wow, so it's not about the age or the strength of the mutant cell itself. It's a lock-and-key mechanism that only exists for a fleeting moment.
Both the winner and the loser must be simultaneously existing within that 48 to 96-hour developmental phase. The biological hardware required to send the death signal and the receptor required to receive it are only manufactured by the cells during that specific transition. At 96 hours, the genetic programming shifts, the hardware is dismantled, and the arena door closes permanently.
Who controls that door? How does a cell's internal clock know it's time to build those receptors?
The timing is governed by specific transcription factors, primarily two known as brachyury and mesodermin.
Let's spell out what those do for the listener.
Sure. Transcripting factors are master proteins. They physically bind to a cell's DNA to turn giant blocks of genes on or off. These two, brachyury and mesodermin, act as the ignition switches for gastrulation. The researchers discovered that if you use genetics to delete them from a cell, that cell becomes completely blind to the supercompetitors.
Because without those transcription factors, the cell's DNA never gets the instruction to build the receptors for the death signal. The mutant is physically touching it, but the biological doorbell has been completely unwired.
A perfect way to visualize it, it proves that the susceptibility isn't a flaw. It is deeply intertwined with the normal, healthy genetic programming of early development.
Which brings us to the ultimate question. We have this highly regulated, strictly timed cellular combat. Why? Why does a developing embryo deliberately subject itself to a purge right at this exact moment?
It comes down to the sheer scale of what happens immediately following this window. Gastrulation is an explosive phase of growth. In a mouse embryo, the core population of cells will increase roughly 100-fold in just 36 hours.
100-fold replication in a day and a half. That is an insane biological boom.
Right, and when you are about to copy a biological blueprint 100 times over to lay down the permanent foundation for the spine, the brain, the organs, you cannot afford a single flaw in the master copy. This competition is a ruthless fitness checkpoint. The embryo uses this window to force cells to test each other's metabolic and structural integrity.
So if a normal cell has even a slight lag in its metabolism,
The supercompetitor senses it, triggers the mitochondrial self-destruct, and seamlessly takes its place.
so guarantees that only the most robust, high-performing cells are allowed to participate in the massive replication phase.
Yes. And the paper emphasizes that this process is phenotypically silent, meaning despite this massive internal war, the resulting blueprint isn't ruined. The gastruloids still successfully form the correct germ layers and structures. The building still gets built perfectly. It is simply constructed exclusively by the victors.
But surely the embryo doesn't want to risk destroying too much of itself. Is there any way to turn this purge off, or does every single cell have to fight for its life?
The embryo actually has a highly sophisticated way of managing this, and it involves the signaling environment. The researchers mapped out how outside factors influence the arena, specifically looking at Wnt and BMP signaling.
What are Wnt and BMP?
They are morphogens. Think of morphogens like a chemical scent sprayed in a room. The cells use the concentration of these chemicals to figure out where they are located in the 3D space of the embryo, like GPS system telling them if they are at the top, bottom, front, or back.
So how do these GPS signals affect the Hunger Games?
The researchers found that artificially flooding the gastruloid with high concentrations of Wnt and BMP signals, which is called agonism, completely suppresses the cell competition. These morphogens alter the internal state of the cells, essentially acting as a molecular shield.
Oh, I see.
Yeah. When bathed in high Wnt and BMP, the normal cell's mitochondria simply ignore the stress signal from the supercompetitor.
So the embryo can use these chemical gradients to designate safe zones.
Exactly. The embryo isn't just running a blind blanket purge. It produces Wnt and BMP in specific geographic locations, like the core of the developing tissue where certain crucial cells are desperately needed. Those core cells are shielded by the morphogens so they can grow safely.
While the cells on the unprotected periphery are left vulnerable to the fitness check.
Right. It is a geographically managed pruning process.
That is just, well, it's staggering. It's an entire architectural strategy playing out on a microscopic level. Let's pull all these threads together for you listening. What we're looking at is a profoundly efficient system where mutant supercompetitors can overtake an entire population, not by starving them for space, but by directly triggering a mitochondrial self-destruct sequence. And this is all locked into a fleeting developmental window right before explosive growth, geographically managed by chemical shields, to ensure the embryo is built from only the absolute highest quality material.
A beautifully brutal, ancient mechanism for ensuring biological perfection at our very genesis.
So what does this all mean for you, the listener, today? Because you aren't an embryo anymore. Your developmental window's closed a long time ago.
Well, this brings us to a really provocative, lingering thought. We've spent this time establishing that the ability for a mutant p53 deficient cell to actively dominate and execute healthy cells is a perfectly natural, deeply embedded program designed for our earliest days. It is a weapon the embryo is supposed to have.
Right, it's a feature, not a bug.
But, as we discussed earlier, in the adult human body, p53 is the ultimate tumor suppressor. When an adult loses p53, the result is cancer. The traditional view is that cancer cells simply grow out of control because their brakes are broken.
Because they just won't stop dividing.
Right. But this research forces us to ask a much darker question. As you go about your day, consider this. Is the terrifying, destructive progression of modern cancer actually an aggressive mutant cell remembering how to pick the lock on this ancient embryonic program?
Oh, wow.
Yeah. Is a tumor just hijacking our own original developmental Hunger Games to actively wipe out our healthy adult tissues?
That reframes how we think about disease entirely. It's not just a breakdown of this system. It's an ancient system being rebooted at the exact wrong time. That is deeply unsettling, but an incredibly fascinating concept to mull over. Thank you for joining us on this deep dive. Keep questioning, keep exploring, and we'll catch you next time.